Okay, so the other thing is that we've seen this diversification generated the in these Stradon eukaryotic systems. Um, are there any other such diversifications that you see in other contexts or in other organisms?
Yes, as you recall in the original or the first paper on the ternary systems, we did this analysis of trying to look at the chromosomal locations in
Right.
actinobacteria which have the linear chromosomes and are enriched uh, in these ternary systems. And earlier studies on actinobacteria, going back, I believe, to Hopwood himself,
Right.
suggests that there is a certain logic to the chromosomal organization of genes in these linear chromosome actinobacteria. Closer to the center, you have a greater enrichment of the core genes, and farther out towards the extremities uh, of the arms on either side, you have the more variable components, especially those involved in conflict, secondary metabolism, and an enrichment of transposable elements. And when we plotted the chromosomal locations of these ternary systems, we found that they seem to lie at the junction between the core region and where the chromosome diversifies, and there is evidence for recombination happening and even chromosome length change in actinobacteria, especially streptomyces, which has been studied. So the fact that there's a very sharp peak, a statistically significant peak on either arm of the chromosome, uh, close to that point where you enter into that more diversified part of the two arms. That's where these ternary systems tend to sit. So what that suggests is that it's a recombination hotspot, and that recombination hotspot is probably exploited in order to dock in a variety of systems which exist on their own elsewhere, but through recombination, a copy of them is docked into the core of the ternary system at those spots, resulting in their diversification. So many convergent mechanisms of recombinatorial diversification seem to have happened. And of course, there is the mutagenic mechanism of diversification, which we'll talk about too.
Right. Right. Yeah. So I mean, that is often in immune systems, and this seems to be convergence. You have various means by which you can achieve um, recombinatorial diversity. We even saw that in the polymorphic toxin systems. This is a fairly
Yes,
well-known way of getting those RHS repeats
yes, yes,
combining in cassettes in and out.
right.
Yeah, yeah. Okay, so I think we still have some ground to cover. So let's. So there were many other very interesting systems that the EADs led us to, and I think we can switch back to that and see the diversity of those systems and the insights derived from them.
Indeed. So by pursuing both Trodden and EAD, we were able to discover a bunch of additional systems. And by pursuing the EADs, like BDLD3, for example, and some of the other EADs which we found earlier, we were able to find systems where they are combined to the EAD, but they also occur in versions where they are independent of the EAD, or what one could call the primitive state, which then got recruited to a larger system through this combination with the EAD. And I think I will group them for the ease of discussion into the broad categories which we found amidst them. And one of those was what we may term the lectin-like domain systems.
Right.
So we found that BDLD3 can be fused to two unrelated lectin-like domains. Nevertheless, they are lectin-like in that versions of those domains bind carbohydrates. One of them is the NPCBM, which had earlier been found in carbohydrate processing enzymes. But we found a diversity of those fused to BDLD3. And in addition to BDLD3, they were also fused directly to effector domains like the TIR domain. Now, in the case they are fused to BDLD3, usually you had an adjacent gene which encoded for a BDLD3 fused to an effector domain, like say the PNPase domain. So you had that effector being brought in by a BDLD3 interaction. So the NPCBM could be connected to the effector domain. When it's fused to the TIR domain, it was a direct fusion. And these systems may additionally feature APA TPases, suggesting that they are ultimately recruited or they are recruiting an apoptosome like complex. And the notable thing about these NPCBM uh, domains was that unlike the characteristic carbohydrate processing enzymes where they may be found, these are for most part intracellular. So, or they may be membrane associated, but on the intracellular side. So it looks as though they are some kind of a receptor. Now, very parallelly, we found the FGS domain again fused to BDLD3. And in those cases, the operon codes for another BDLD3 protein which can get in an effector. And in the same vein, you could have other EADs, for example, EAD8, which we found fused to the FGS domain. And this FGS domain, we encountered it earlier in the ternary systems where it can occupy the effector position in multiple ternary systems, for example, fused to the C termini of the VWA domain in the VMAP ternary systems, which we talked about the last time. And we also saw that these FGS domains, which have this C-type lectin-like fold, were originally seen in the Müllerian DGR systems, which are diversified by the reverse transcriptases. And even in these cases, where we found them fused to EADs and BDLD3, we did find evidence for the genome containing the diversifying reverse transcriptase, along with the template strand, which is used to diversify the C-type lectin domain, which is this FGS domain. So in some cases, that reverse transcriptase is embedded into the operon just adjacent to it. And in other cases, it may be a little further away in the genome. But it always has that template with which it diversifies these. And apart from being fused to BDLDs or directly to effectors, it may also be fused to one group of ATPases, which we discovered many years ago, called the CARP NTPases. These CARP NTPases had been implicated in phage resistance, but little else was known of it. And we showed that several conflict systems were based on this CARP NTPase, which we now know is likely to be a flanking sister group of the stand-like NTPases. So it occupies a configuration or a position very similar to the stand-like NTPases. So in place of the NARCT or APA-TPase or one of the new stand domains which we discovered, which may show a fusion to FGS, you may have, or to NPCBM, you may have this CARP NTPase, which is directly fused to the FGS. So we have two unrelated lectin-like domains, which are either directly fused to effector domains or are likely recruiting an effector domain via an EAD death-like domain. So what is going on here? A clue for this came from the well-studied lectin-like domains, which are the spry, pry, neuralized like domains. These are well-known lectin-like domains in metazoans and other eukaryotes, which have been implicated in immunity. For example, one of the immune mechanisms against HIV, an important immune mechanism against HIV-1, I believe, is a protein with the spry-like lectin domain, or the spry lectin domain, which is fused to N-terminal ring type E3 ligase and D-box domains. TRIM5, the human TRIM5, it's a restriction factor against the AIDS HIV-1 virus. It specifically ubiquitinates components of the invasive entity and thereby allows an immune response, intracellular immune response against it. So we propose that this mechanism is a far more widespread and a generalized mechanism which had its roots in the bacterial world. And that's what we are seeing in the form of these NPCBM and FGS domains, where the FGS domain, where either of these two lectin domains bind the invasive entity, either because of a certain glycosylation feature, if they retain the classic lectin capacity, or they may be simple protein binding domains. So in a sense, they are antigen receptors or invader receptors, which then activate the fused effector domain to either trigger cell death or launch an attack on the invasive entity. And this could involve an EAD-EAD interaction or a death-death interaction. So the discovery of this lectin-like principle as a common mechanism, which has swept in a wide range of lectin-like domains, both in eukaryotes and prokaryotes, such as NPCBM, FGS, and the spry-like group, spry, pry, neuralize, discoidin in some cases, is an important aspect of immunity. So this is a very generalized principle of the immune mechanism of recognizing the invader through a lectin-like domain.
Yeah, and even the thing that the spry domain is found in bacteriophage tails, if I'm not wrong.
Yeah, so that is an interesting offshoot that the FGS domain was in Miller's original systems. He found those in those bacteriophage tails, which diversify through those DGRs to increase their host range. And likewise, the spry domain, which is found in the eukaryotic TRIM proteins, which are part of the antiviral immune system, they have their roots in bacterial tails, bacteriophage tails.
Right.
So that, and we described some of these in that work on the jumbo phages too.
Right.
So this was one theme which we uncovered. The other rather remarkable theme, and I think this is the first time any such theme has been reported, is a conflict system which directly acts at the level of the RNA polymerase. And we ran into these by looking at the associations of EAD10.
Correct.
And EAD10 can be fused to things like the caspase domain. And then one of the things which we found it to be fused to was this remarkable set of domains, which has the following architectural theme. So there is a constant part to these. And this constant part going from N terminus to the C terminus is a string of TPR repeats, followed by this GRI-ABC domain, and then a pin domain. So just to unpack this a bit, what is this GRI-ABC? So these GRI-ABC domains, they were first characterized in the context of transcription in prokaryotes, and they are things which associate with the RNA polymerase. So it's a FKBP, the peptidyl prolyl isomerase like fold, which is found in the two transcription elongation factors, GRI-A and GRI-B. So it's the C-terminal domain of theirs. Hence, we call it the GRI-ABC, that is the C-terminal domain. And in the regular transcription elongation factors like GRI-A and GRI-B, uh, this domain, it engages with what has been termed the secondary exit channel, which is a nice pore-like structure, which we have studied quite a bit in our other investigation, including identifying that R modes domain as its core, which is formed by the beta prime subunit of the RNA polymerase. And that is the channel through which the nucleotide substrates diffuse uh, in and out of the RNA polymerase reaction chamber, so to say. And the 3' end of a transcript, if there is stalling, can be backed up into that. And this GRIAB N-terminal part can extend into that secondary channel. It's a long coil, and it can engage that backed up transcript and allow its cleavage by using the very same active site of the RNA polymerase. And then the transcription can resume. Now, there are some variants of this theme where there are proteins with just this GRI-ABC domain, which can act as transcriptional inhibitors, which instead of acting as an elongation factor, they can associate with this exit channel and inhibit transcription. But what we found here was rather remarkable in that this constant region with the GRI-ABC domain gets flanked by these TPR repeats and the spin domain. And as we discussed a couple of episodes back, the pin is a metal-dependent endo-RNase. So in all likelihood, it is cleaving the transcript at the level of the RNA polymerase itself. That is, even as transcription is happening, this system is likely to cleave the transcript. Now, there is a further twist in these architectures. N-terminal to the TPR repeats, there is a remarkable diversity of effector domains. Or there may be EAD10, as I mentioned, and that EAD10 could in turn recruit other effector domains like the caspase domain. So it's the usual collection of effectors, TIR, HNH, the REase, the restriction endonuclease, the PNPase, and trypsin-like peptidases, SIR2. That's a remarkable diversity of these, which we find in these at the end termini of these GRI-ABC systems. And what we propose is that these are conflict systems which are working at the level of the transcription. And perhaps those TPR repeats serve as some kind of a bridge between that effector and this core module with the GRI-ABC and the pin. And the GRI-ABC can engage, like the regular GRI-ABC, the RNA polymerase exit channel. And if there is a sensing of an invader, which may happen at that juncture between the TPRs and the GRI-ABC domain, then the pin domain cleaves the transcript. Thus, it could limit the transcription of viral proteins. Now, if the virus, as we know, many viruses, they transcription of viral genes, not proteins. So we know that many viruses, they have inhibitors against the host conflict systems. So what we propose is that if there is a viral inhibitor which blocks this core system, then this N-terminal effector is unleashed, which is essentially an apoptotic effector which brings down the cell through an apoptotic mechanism. So that's like the final option to bring down the cell if the first line of defense fails. And that explains why the N-terminal effector domain is very variable, whereas the PIN, it's a novel version of the PIN domain that remains quite constant. So the idea is that there is an arms race going on with these viral inhibitors. So you need different kinds of effectors to evade the viral counterattack. So this is a very remarkable transcription level conflict system, the type of which has never been described before. And one could even imagine some biotechnological applications for something like this to target genes which are being transcribed using this kind of a molecule. And finally, by pursuing these EADs, we found yet another class of conflict systems. And these are based on GTPase of the general RAS, Rho, RAC like group, the small GTPase superfamily, which we had first recognized in metazoan immune systems or apoptosis systems, which in our old paper on apoptosis, we had called APGTPase, or the depth GTPase domain. And these APGTPase domains in eukaryotes, they occur like in that large protein with ankyrin repeats and a kinase domain, where the kinase domain acts as the effector in the eukaryotic APGTPase system. But its exact origins had remained quite mysterious. So this one, just to be specific, is the DAP kinase. So the DAP protein, which is a well-known apoptosis effector. But here we finally have the prokaryotic roots, and we can show that this eukaryotic apoptosis protein is just a little, it's like the tip of the iceberg of a vast prokaryotic immune mechanism. So in prokaryotes, these APGTPases have a very characteristic architecture. They're flanked on one side, again, like just to bring the comparison with the TRADN domain, they occupy a significant central position rather than being at the end. So they are flanked by globular domains on either side. And most often at the end terminus, they have LRR or leucine-rich repeats. In some cases, instead of leucine-rich repeats, they may have beta propellers, and in a few cases, VWA domains. And at the C termini, they have a highly variable region, which may feature one of many effectors. Again, our usual suspects like REases, TIR, caspases, PNPases, so on, all the whole range of usual effectors. Some cases, it may even be other stand domains like the NARC. So the idea here is that this is a one molecule immune system where the LRRs recognize the invasive entity. And there are lots of parallels for it, like the plant immunity APATPases, where the LRRs recognize the invasive entity, or in an extracellular context, like the VLRs of the jawless vertebrates or the TLRs of various metazoans. So the LRR is the sensor which senses the invasive entity. And the APGTPase in this case is a switch which in all likelihood acts very much like a RAS or Rho or any of those small GTPase like switches, which turns on the C-terminal effector, which can then mediate an effector action either through direct cell killing or uh, action on some other molecule and preventing the invader propagation or acting on the invader molecule itself. The types of effectors which we see point to all these possibilities. For example, one of the effectors is a CNMP cyclase, suggesting that a cyclic nucleotide signal is first sparked off. In some cases, the effector is a caspase, which could mediate a proteolytic cascade or directly lead to cell death. Likewise, it could be with TIR and DR hide, REAs, and the like. And in other cases, it could be more indirect. Um, for example, when we have a NACT, it may trigger a further round of signaling. So these were some important themes of new conflict systems that the EADs led us to. Now, on the other hand, the trodden domains also led us to some new systems, and there was some overlap between the two. And I'll hence only mention the unique thing which we got from the trodden side, which was these GT4 systems. Now, what are these GT4 domains? These GT4 domains are glycosyl transferase domains, which have a double Rosman domain catalytic module. So there are two Rosmanoid domains, tandem ones, which constitute the uh, active site of these glycosyl transferases. And they are glycosyl transferases which modify a wide range of substrates. And what we found was that there is a large number of these which are fused to the trodden domains in especially in nidarians like Stylophora and some other corals like Acropora. So in these other corals like Acropora, they can be fused to caspases, depth, NACT. So there's an extraordinary diversity of architectures again, and huge numbers. I think there may be up to 500 of them in some nidarians. So 500 paralogs. It's a gigantic LSE in one of the corals, Acropora. So and there's a parallel diversity of architectures along with the expansion of the glycosyl transferase domain itself. Now, in the prokaryotic world, the same GT4 can reappear in the effector position with the APGTPases or with APATPases and the N-stand domains, sometimes with NACTs. And we had earlier encountered this in certain systems, conflict systems, more of the polymorphic toxin type, which are secreted through the type 7 secretory system in actinobacteria, which are some of the largest proteins which we know, gigantic proteins with a whole string of effector domains, which seem to be secreted out and deployed in interorganismal conflict. So again, the GT4 glycosyl transferase is an effector which is shared by different kinds of conflict systems. Just as an aside, it's an infrequent but still present effector even in the VMAP ternary systems. So what do we think this GT4 is doing? By virtue of being a glycosyl transferase, and we for most part see these paralogs to be active versions, we think that it is a mechanism which is parallel to the lectin-like domains which I just mentioned, wherein these transfer a sugar moiety or more onto either self proteins or to proteins from the invaders. And this glycosylation of proteins breaks up interactions of the invasive entities and cellular factors, or it triggers a cascade of cellular interactions which may lead to apoptosis or easier recognition, uh, in a sense, sort of like opsonization of the invasive entity, but in an intracellular context, leading to its destruction. For example, there are ubiquitin ligases which have domains to recognize glycosylated proteins. So this could make them good substrates for being ubiquitinated and thus cleaved. Thus, the study yielded a whole range of immune mechanisms. And importantly, it told us that this process of adaptive immunity, which was thought to be a unique premise of the metazoans, especially the vertebrates, probably has much deeper uh, roots or a much wider spread, even though it has convergently evolved in each of these cases, like the FGS domains of and the FGS-based systems, the great diversity of them in prokaryotes, are likely some type of adaptive immunity, working through a mutagenic process with the reverse transcriptase, uh, in ways parallel to the mutagenic diversification of antigen receptors in vertebrates.
Mm-hmm. Yeah, the other point, I know we've been speaking for some time, but the other point that actually comes out well, and I remember this point that you made when we were discussing this, is, why do we see in ternary systems, why do we often see very distinct types of stand domains? And also there was this, and there was also a type where there was this MNS type stand domain, and then many distinct types of stand domains in ternary systems, sometimes inactive versions, and how they, why their role as in nucleic acid interacting um, proteins allows them to even function as receptors. I think you had a fairly clear understanding of that.
Right. So that was another important offshoot of this work, that the stand has gone in two completely different directions. And this harkens back to our original understanding of the stands, that all stands have ultimately descended from the CDC6 ORC like proteins.
Right.
And that the more basal branches like the MNS group, uh, they are the transposon versions of the CDC6 ORC complex, where they work in an equivalent role, but in the replication of these transposons or phages like mu.
Right.
And thus, their ancestral function is forming a toroid which associates with DNA and binds DNA. So, in a sense, they can act as DNA sensors, parallel to what we see with the helicard-like proteins or the helicase nucleic acid receptors in metazoans.
Mm-hmm.
So they have that innate potential. On the other hand, being A plus ATPases, uh, they have this capacity of serving as reorganizers or remodelers of complexes or switches. So I think both these functions have been utilized in parallel ways, and they represent two completely different directions the stands have taken. So the great diversity of stands in these systems on one side represents their use uh, as NTP dependent switches, and that continues to the apoptosome, the inflammasome, and the like of eukaryotes. And we can see that the eukaryotic versions are merely a small slice of the extraordinary diversity of these, which we see in the prokaryotic world, where they act as switches which unleash a variety of effectors uh, and the like. On the other hand, the key finding from these two papers, which we just published, is that there are inactive stands, the two I stands, as we call them, where the catalytic motifs are completely gone, but still they are a very key component of two of the types of ternary systems. They are the third component of the ternary system. So, what is an inactive stand doing there? And that's how the hypothesis came about, that these are nucleic acid receptors. They are functioning there in the capacity of nucleic acid receptors, and thus they act as the sensory components. As we discussed the last time with these ternary systems, one of the components has a sensory role in sensing the invasive entity. So these I stands or the inactive stands could function as these sensory components which bind nucleic acid. And indeed, that nucleic acid binding function is retained in multiple ways, because even with these AP GTPase based systems, with the GT4 based systems, we found some prokaryotic GT4 deploying systems which featured a novel inactive stand. Again, supporting that there have been at least 3 different inactivations of stands which are used in these conflict systems. And we see that some of those stands, like in that new system which we found, which deploys GT4 amidst many other effectors, are related to these more basal branches rather than the crown group with NAFT and AP-ATPase. So they are closer to the base where the CR effector stands and MNS and the like inhabit, the more basal branches. And amidst those are at least 2 RNA binding versions, one of which is definitely known to bind RNA, which is the S29 or DAP3 ribosomal protein.
Mm.
And this is a stand NTPase which is found right in the ribosome. It has a GTP binding, that is, it's a GTPase rather than an ATPase. And it bound single-stranded RNA as a monomer in the ribosome. And interestingly, it also has an apoptotic role. Even though it has this core ribosomal role, it can, under the right conditions, trigger apoptosis uh, as part of association with that ribosome. So we see that these two functions are intimately connected, and some of these basal stands could serve as, which have become inactive, could merely serve as a nucleic acid receptor, even as the DAP3 stand binds RNA in the ribosome. The other one, which is less studied but could play a similar role, is the RNA12 protein, which is another stand domain protein, which very poorly understood, but at least our prediction would be like S29 or DAP3, it might be binding RNA and playing a similar role.
Right, right, right. I think you've covered most of, I mean, almost all aspects of these two novel discoveries. But I think there are some general points that have emerged from this that are beyond this. That is the general aspects of evolution of immune systems themselves, um, and perhaps we can sort of now go to some more general points that have emerged from these studies.
Yes, I think we can sort of round it up with that. On one of those general principles is that there is a repeated drive for different mutagenic mechanisms to be incorporated for diversification, either through deaminases, which came from a very different source in the vertebrates, or through reverse transcriptases, or through the terminal transferase, which is seen in vertebrate immunity, one of the Pol beta like nucleotide transferases. So there are many different independent mutagenic mechanisms, but there's a repeated selection for mutagenic mechanisms in immunity. And these are often coupled with an apoptotic component that if immunity fails, you bring down the cell through apoptosis. So there's a very intimate connection between the two. And then there is this principle of intercepting the invader either within or without. And the prokaryotic immune systems have an extraordinary diversity of internal interceptors, as we described a whole lot of them. Whereas that is there in several eukaryotes, like the plants base a good part of their immunity through this mechanism of internal interception. But animals, especially vertebrates, you see a strong tendency for intercepting the invader on the exterior. Now, in these multicellular prokaryotes, we see that a tendency towards that, like the EACC1 system, which we described, which has an extraordinary diversity of intracellular effectors, but the key sensory component, which is likely to intercept the pathogen or recognize the invasive entity, that happens in the membrane. So as you move towards multicellularity, when you have an ensemble of cells, a greater need for surveillance at the surface seems to emerge. And that may even be seen in the I-stand-2 systems, I-stand-1 systems, where there's a dominance of those membrane-associated components. The DOGTPases are again membrane-associated.
Right.
Or they have a dominance of membrane association. So multicellularity may have gone hand in hand with a greater move for coordinating interception on the exterior in a more immediate way with reacting to the pathogen. With MAX, we describe these systems with the Schlafen-like endoRNases, which are fused to a peculiar version of the histidine kinase domain, which are often coupled with polysaccharide modification systems. And that points to, again, an internal immunity with an external immunity. But that external immunity in most bacteria is not very immediate. So you keep changing your polysaccharide when you're under the phage threat, or you keep changing it at some frequency so that you have a chance of evading the phage threat. And if the phage gets past it, then you may activate this internal Schlafen-based endonuclease system, which may either intercept it internally or kill the cell. And indeed, we have some other systems like that, which we think might operate on both sides. But here, these dedicated systems which may intercept things close to the membrane suggest that with multicellularity, you have a more immediate need of extrinsic interception. And finally, one could say that this is revealing something very remarkable about the linguistic nature of these architectures, or linguistic principles behind these architectures. So what we are seeing is that we have the same kinds of domains they have established these apoptosis and immunity systems in multicellular bacteria and in eukaryotes. Now, they are homologous domains. So there has to be a causal evolutionary link between the origin of apoptosis and the acquisition of these domains. So one conjecture there would be that the origin of metazoa was very clearly related to the bacteria which they associate, which their ancestors, the stem metazoan lineage associated with. And this is not just any multicellularity. One could argue that there was a much deeper multicellularity, perhaps in the animal fungus ancestor, or even going back at some incipient level to their common ancestor with the slime molds and amoebozoans sort of switching on and off between multicellularity and unicellularity. But metazoan multicellularity has these very unique components in the form of these apoptotic systems, which clearly distinguish them even from their sister lineages, like the choanoflagellates. As we have been discussing, that many of the students of animal origins ignore this important point.
Right.
So, what it suggests is that it was not just the coloniality of choanoflagellate like organisms, which was sufficient or the only factor which gave rise to animal multicellularity, uh, but a combination of other things uh, which came about. One of which was perhaps association with cyanobacteria or some other bacteria with the active calcium signaling, perhaps in a stromatolite-like environment, like what we discussed by studying the calcium signaling systems with Daniel Schaefer and Max uh, a few years back. So that pointed to a presence of a repeated association with cyanobacterial uh, uh, symbionts or cobionts. And now this apoptosis system shared with multicellular bacteria suggests that the association of some choanoflagellate-like organisms, that is morphologically choanoflagellate-like organisms, of the stem metazoan lineage with uh, multicellular bacteria also seeded them with these apoptosis genes or apoptosis domains, the domains of death, uh, which allowed solidification of their multicellularity based on the principles we discussed in the very first talk. And
Aravind, but this is one point that still bothers me right from the for the past 20 years, and I think perhaps you can clarify this a little more. There is,
hmm,
at some point there was this idea that perhaps apoptosis was somewhat ancestrally present in plants, fungi, and animals. Of course, that was based on a tree with very little data. That is in the eukaryotic phylogenetic tree. It was just developing then. But um, I guess we can say with some degree of certainty that this event has occurred at least on three independent occasions. That is the transfer of the apoptosis complex of genes into fungi, uh, where it is used for anastomosis, in plants, where it is again part of an apoptosis like response, and in metazoans.
I think we may even add some further nuance here that there
Right.
are that both are valid at some level. So there
Mhm.
were some early inputs,
Right.
like just acquiring stands and APA TPases, which may have happened early on and provided an incipient level of apoptosis.
Right.
And at least looking at, say, the NARCs like HET-E, the HET-E genes, which are NARCs,
Right.
that's the H in the NARCs, and
And the telomerase.
the telomerase, importantly, the telomerase subunit TLP1.
Right.
That's an even earlier acquisition.
Right. Right.
So there have been multiple, the telomerase acquisition was a very ancient one. And then there were reacquisitions independently, like HET-E, the HET-E genes, and then the NOx proper, the NLRs of the metazoans. So there were reacquisitions. So it could have been that the common ancestor had that incipient version through the earlier acquisitions from bacteria. But it was reinforced depending on the movement towards greater or lesser multicellularity in a given lineage.
Mm-hmm.
So there is this constant movement back and forth between multicellularity and unicellularity, so to say. And if you move towards greater multicellularity, there may be an advantage for having more of these apoptosis systems which have evolved in the bacteria. and that reinforces that multicellular state. And this relates to this point which I keep calling the linguistic principle. So why is it that the same domains are involved? So I see these domains as having some kind of a linguistic structure to them, in that they come as properly conjugated at the first stage, at the first step. So what I mean by conjugation is like in language. So if we put an assembly of words there which are not in their root forms, that is, especially in a language like Samskrita, this is very good to demonstrate, where you can have nouns and verbs in their root form. Uh, so if I just throw in some verbs there which are unconjugated, uh, even German works quite well for it. English is not that good. Uh, so if I just put in words like to run, stone, so on, some collect conglomeration of words, and because English doesn't inflect so much, even as much as its cousin German, it's very bad to demonstrate in English. Of course, it's best demonstrated in Samskrita, which conjugates and inflects very in a very detailed way. So if you just put in the root forms, sentences won't form if you mix them together. But if you have them pre-conjugated and you mix them together, there's a high chance that good grammatical sentences form when you randomly draw out these words. So that's how I imagine some of these domains as having an intrinsically conjugated state, uh, like the pre-conjugated words. So the stand NTPases, the caspases, the trypsin-like peptidases, I see these as part of a vocabulary which tends to come as pre-conjugated. So if you throw them together, there's a good chance that they form a grammatical sentence, which in the biological context would be a meaningful system,
Right.
a functionally meaningful system. So there are some other domains which have much less of a tendency to come in a pre-conjugated form because they are stuck in some very unique configurations, which may not be amenable to any kind of use in other contexts. So I think this apoptosis system in prokaryotes, it evolved, it's by virtue of its very origin in the context of conflict. There was a selection for those domains which could come in this pre-conjugated form. Because if you are able to form meaningful sentences or meaningful domain architectures when we talk of proteins, which can confer function right away, then they are more likely to be exchanged through lateral transfer and fixed. Uh, uh, that is more likely to be fixed post lateral transfer. So they can be ported between one organism to another, because if two of them come together because of that pre-conjugated property, they would form a meaningful sentence and thus provide a function. Uh, so that allowed them to be transferred to eukaryotes too in the same way, and now seed a new function in eukaryotes.
Right.
Now, uh, on the other hand, another aspect of this linguistic principle are these EADs and death-like domains. So that very EAD principle, it is like providing a conjugational hook. So again, this is best illustrated in a good language which inflects well. So if you just give the root form of the uh, of the word, you are not going to get meaning out of it. But if you give the root form with a certain ending or a conjugation or an inflection, then it is going to find the appropriate word. So there will be a certain syntax which automatically is determined by those inflections and conjugational markers. So you can see these death-like superfamily domains and EADs as these conjugational markers or inflections of nouns. For example, in Sanskrit or in German, certain endings will tell you it is the accusative case. So it will exactly go and fit as the object of the sentence and not its subject.
Correct.
Whereas something else will fit from its inflection only as the subject.
Correct.
Likewise, the depth and the trodden domains uh, come with that kind of an inflection which can fit the domains around them and themselves into an appropriate grammar. So I think this is the most general evolutionary principle which we can glean out of this, the linguistic principle, and that it's a very important thing in biology, which sort of acts as an overarching Platonic principle above the low-level action or the blind watchmaker action, to use Dawkins' phrase, of natural selection.
Right, right, right. And the interesting thing is that you have this conjugated vocabulary that comes together, but then the effectors again can vary greatly with
Yes,
so you have BCL2 and
yes,
side and so on, which is very
right,
distinct.
right. And that is again explained by this linguistic principle. Uh, like, say you make a sentence that Ram went,
Right,
and you just leave it like that. You can add after that went, Ram went to school,
right,
Ram went to the office,
right,
Ram went out of his house.
right,
So, in a sense, that core part is like that first part.
right, right,
And the effectors are like everything else which you can add with very little effort.
right,
And the EAD, like say it needs to be in the accusative case, say the EAD would be like that accusative ending,
right,
which you add to the noun. And then it can fit in that accusative position with any other, whatever is the preceding sentence, which is supplying the rest of it.
right. That's a fascinating analogy. I mean, yeah,
Yeah, so I think that it's a very good illustration of that the underlying linguistic principle in biology.
right,
In many ways, the linguistic principle is sort of an analogy to Euclidean, or rather than analogy, it's a parallel mode of understanding the universe to Euclidean geometry.
right,
Of course, a physicist may talk about Riemannian geometry, but for most purpose, we live in a Euclidean space. Our common sense is in a Euclidean space. Uh, so we can, so the constructs which we use, the axioms which we use in it, help us to make sense of the world, especially the world of geometry, the world of physics and chemistry. They all stem from Euclidean geometry. In a sense, much of basic biology too, that is globular proteins and interactions of globular domains, the central dogma, they are all consequences of Euclidean geometry. But there is a parallel principle, which is this the linguistic principle, which is another mechanism of apprehending knowledge.
right, right, and yeah, it's again a sort of two old world ideas, one linguistic based and one geometry based. That is of the Greeks and the
Yeah.
ancient Hindus, coming together in this kind of
Yes.
interesting way that you presented.
Right.
Yeah, yeah. So, and I don't know. I mean, I have one other that is we can actually end it very shortly. But I remember somebody somewhere commenting that we have all this big alphabet soup of domains that are just names. But I think when you talk about languages have many words too. So in order for one to be proficient in a language, one has to know the vocabulary. And
Exactly.
you
So if
know,
somebody talks of alphabet soup, I always tell them one thing. Any technical discussion would always sound as an alphabet soup to the outsider. if you are serious about a topic, you have to get acquainted with the language in which it is spoken. There are no two ways about it.
Right. Right. Yeah. And yeah, fascinating discussion. I think, whereas for many people who are listening to this, the series of talks would sort of discussions would give them an overall picture of how we arrived at these discoveries and the time and the kind of different aspects of the hypotheses and thinking that we had to have to reach this. But it also, I think, for people like me who have been insiders, this has been a great way of, in a sense, revising the vast amount of work that has happened outside as well as within our group, because it's not often that we actually sit and discuss the whole of it together, um, in this fashion. So I think even for me, it was a fascinating recap of everything, and I'm sort of refreshed. And I guess we need to proceed to look for more interesting systems like
Yeah, I agree. These are perhaps the most interesting systems we have ever discovered to date. Personally, I think the systems which we uncovered in these two papers are the most exciting which I have come across in my existence. And it also closes this chapter on the origin of apoptosis uh, in a fairly comprehensive way.
Yeah. Okay then, Arvind. I think
Okay.
hopefully
Yeah.
until say the next time when you feel like doing this for some other topic, we will sign off for now then.
Yeah.